Hi, everybody, and welcome to my master's thesis oral defense. Like Dr. Weber said, my name is Kate Tillotson, and I had the distinct pleasure of doing my work in her lab here at SIU. So I'm going to walk you through the two different chapters of my master's thesis, one on the pollinators of Triadinus, and then the other on a citizen science project that I employed here in the plant bio or at SIU. And I'll talk a lot more about that. That'll be my second chapter. So we're gonna go into my first chapter, which was an assessment of the pollinator assemblage for the genus Triodanis and a look at the breeding system variability. So in the introduction and background, we're going to just introduce reproduction in flowering plants, because it's really awesome. We're then going to talk about some of the pressures on these interactions, both contemporary and past. And then we're going to look at some of what we know about the natural history of Triodanis and what makes it a really awesome system when you're looking at reproductive strategies in flowering plants. We're going to spend a bulk of this presentation talking about my methods. And through my methods I'll also walk you through my analysis and some of my results. And then we'll wrap everything up, some key takeaways and some really interesting facts when I was processing this data and what came from it. So we'll jump right in. So when we're talking about floral evolution or the evolution of flowering plants, we can't not mention pollinators. This coevolution is a driver of the biodiversity that we see today. And it's really weird, to depend on a completely unrelated organism for your reproduction. And so, like I said, this has driven diversity in floral forms, and you can just see some of this diversity in the family Campanulaceae, which Triodanis is part of. Okay. So we have this relationship, but it has changed over time. There have been different pressures on the relationship between plants and pollinators in evolutionary history. One of these being dispersal events. So as flowering plants shift their ranges, they may shift into habitats where they have either absent or unreliable pollinators, and this can change their reproductive strategy. There have also been global shifts in climate in geologic time, and this can change the phenology or the timing of life history events in both plants and pollinators. And so if we have these changes across a landscape, we have local adaptation for species, and this can drive species divergence. So we have these past pressures, but now we have contemporary pressures on plant-pollinator interactions. One of the most worrying of these being anthropogenic caused climate change, which has, within the literature, has changed plant-pollinator interactions in really unpredictable ways. We also have widespread human disturbance and habitat fragmentation, urbanization, and then the decline in pollinators overall because of widespread pesticide use in agricultural systems. So we've got Triodanis. We use this genus as a study system to answer ecological and evolutionary questions generally. It has 7 species distributed through both North and South America, It's a weedy annual plant, which makes it great because we can go out and collect it because it's a least concerned species. It exists in a gradient of habitats. So we find it in both natural and these disturbed areas. And it often occurs in sympatry, or it co-occurs. So you'll find a population with several different Triadinus species often. But what makes it really special is that it exhibits this mixed mating strategy. So I think generally we think of reproductive strategies in flowering plants. We think about attracting pollinators. But like I said, there are these pressures on flowering plants, and sometimes they do not have reliable pollinators. So to persist, some flowering plants have evolved a mixed mating strategy where they outcross and they also self. And so we see that in Triodanis. Triodanis has these pretty purple rotate flowers with 5 petals, but they also produce closed flowers that never open. These flowers are obligately selfing, so they are not open to pollinators. They do not go through outcross. And what makes them really awesome as a study system is that the seed capsules of these flowers are morphologically distinct, which means that we can count these seed capsules on an individual plant and look at the allocation of resources to these more expensive open flowers and these resource cheap flowers that are closed. And so we have done that in past studies in the lab. Dr. Bethan Saldi quantified breeding system variability in Triodanis perfoliata over the landscape and found that the breeding system was highly variable and tied that to abiotic factors on the landscape. But for other species in the genus, this breeding system variability has not been quantified. Which leads me into my research. Like Dr. Weber said, we were really unsure if Triadinus even had pollinators. It's this highly selfing genus, or it has species in the genus that are highly selfing. And so it may not really try to attract pollinators, which is sort of like an anthropogenic spin on it, but it may not create rewards for pollinators to attract them. So we needed to establish baseline that there were pollinators. So spoiler alert, there were. And so now we can jump into some of the other questions that I asked. The composition of the pollinator assembly is what I'll be addressing throughout the rest of this presentation. Do pollinators differ between Triadinus species? For this, we found no, not really. I'm going to talk about the pollinator assemblage across the 4 different species of Triadinus that we did sample. And if there is variability, is it linked to breeding system variability? And for this, we found also not really. They shared pollinators across the 4 species. And then does pollinator assembly differ spatially across the range sampled? And that is what we're going to talk about for the rest of this presentation. So to jump right in, we collected 126 individuals on 4 different species of Triadinus, and all of these pollinators fell into 2 functional groups. So if you're not familiar with functional groups, they're just a way of broadly categorizing pollinators that share the same functional traits. And these 2 functional groups were flower visiting fly species and solitary or eusocial small bee species. All of these pollinators that we collected were generalist pollinators across both flies and bees. And if you look in that box, there are other arthropods in there. And we collected all floral visitors on Triadinus. So there are, but that is for a different study, and it did not factor into my work. So to start, we sampled in, we started sampling in Texas because we were tracking the peak flowering phenology for these plants. We then moved northward into Missouri and then west into Kansas, and then finally back east to Illinois. For our standard methods, we sampled across 3 days generally, and we broke our sites into 1 meter by 1 meter microsites because of the patchy nature of Triodanis populations. We sampled at 3 different times of day so that we had temporal coverage across a day to look at any variability. And we didn't sample any site twice in 1 day. So we rotated between sites. When we were sampling, we also observed and characterized floral visitation or habit. And then we caught these visitors for identification. But we did this observation because we wanted to make sure that we were only catching floral visitors that came into contact with Triodanis reproductive structures. We then also characterized local habitat and weather. And we characterized sites on a gradient from highly disturbed to human-maintained natural areas. But Triodanis is really funny. It really likes disturbance, or some species really like disturbance. And so it grows in these habitats where standard sampling is just not feasible, like on the side of a road or in sidewalk cracks like you see here. And so for the best coverage within our field season, we also did haphazard sampling. And now that won't be featured in my results, but I wanted to note that it was something that we did just so that we had the fullest coverage possible. At sites, we also characterized co-flowering species, and this was just a way for me to sneak in all the beautiful flowers that we saw at the Stengel field site. And that's actually where I'm going to start. We started our field site here at Stengel Lost Pines in Smithville, Texas. But before I really jump in, I want to go ahead and note some of the things that you're going to see when we're looking at sample sites. The title will either have a green box or a red box, and this indicates a human preserved natural area or a highly degraded or disturbed area, because we did standard sampling in both. It will also have all of the Triodanis species present at that site. And then while we took a series of diversity indices, we just have some listed here with abundance across genera and genera richness. And then we also have Shannon-Weiner diversity index. For this diversity index, it can fall on a scale of 1 to 5 and generally falls between 1.5 to 3.5. So you can see here that for this diversity, we're falling below the average here at Stengel. Also, slides will have the most abundant pollinators pictured, and flower visiting fly species will have an orange circle, and small bee species will have a yellow circle, just in case you're not familiar with those species. It will also have all of the species of pollinators that we collected at those sites, and asterisks that indicate abundance. So back to the Stengel Lost Pine site. Here was the only site where we found Triodanis lamprosperma and were able to collect pollinators there. The Stengel Lost Pine site was really interesting because it was the first time that we got to observe pollinators and observe their habit. And what we found when we were observing these pollinators is that they were being visited, and pollinators were exhibiting floral constancy. So floral constancy is when on a foraging bout, pollinators are visiting one flower and then visiting another flower of the same species or related species. And so what we found is that on foraging bouts for Hemipenthes sinuosa, or the sinuous bee fly, they were showing floral constancy between Triodanis. But when Triodanis species co-occurred in our sampling sites, they didn't differentiate between Triodanis species. So they were visiting these open flowers, but they weren't differentiating between species, which leads us to believe that these species are sort of creating the same floral rewards across species for these pollinators. Let's see. Oh, Hemipenthes is really interesting because there's just not a whole lot of data out there for these bee fly species, but they are covered in these hairs. So if you're looking for a study system, you could study Hemipenthes because they haven't been studied well as effective pollinators. But I wanted to note that they are these hairy little flies because we also had flower flies that were a bit different morphologically. We then moved northward to Missouri, or Rolla, Missouri, and sampled at the Bahizian Conservation Area, which was a human preserved natural site. Only Triodanis perfoliata was present there, but pollinators were highly abundant here in comparison to some of the other sites sampled. However, diversity was very low, so we had a high abundance of one or two species, really just Ceratina dupla, which is pictured here. We also sampled in Rolla. We also sampled a different site in Rolla, Missouri. So we sampled at Fort Leonard Wood with the help of field managers at Missouri S&T. Biflora and perfoliata were present there, and abundance was similar to what we found at Stengel. You know, this site maybe was a little less degraded than some of the sites we're going to see coming up. While we did sample many different bee species there, the most abundant pollinator was the syrphid fly species Toxomerus marginatus and Toxomerus geminatus. So when I talked about Stengel, I noted that Hemipenthes has these hairs that could collect pollen. But Toxomerus has no hairs present and is a wild generalist. It's pretty well studied that it doesn't show much floral constancy, and that was what we observed when we were observing our pollinators, is that Toxomerus would visit our Triodanis individuals and then would visit other co-occurring flowers in the area. We then moved westward to Konza Prairie. Konza Prairie is a long-term ecological research facility, and we sampled both Leptocarpa and perfoliata there. It is a human-preserved prairie, so humans have reintroduced native grazers and fire onto the landscape, and this is generally not present at the sites that we sampled. While there were bee species present and Agachlorella aurata was abundant, Toxomerus marginatus, again, was the most highly abundant pollinator there at that site. And we have my friend Ben helping me select pollinators there. In contrast, we also sampled in Manhattan, Kansas, where Konza Prairie is located, at a cattle pasture, so non-native grazers. And this was Jeff Taylor's cow pasture. He is the botanist at Konza Prairie, and without him, we would not have found any of our Triodanis populations, because apparently that year was just really bad for Leptocarpa and Perfoliata. So we sampled Triodanis perfoliata here in this degraded site, and over our standard sampling, we only found one individual, which, if you think about it, is really boring to look at these sites for 3 days and not find any pollinators. And pictured here is Sarah Lukowski, who is not present today, but who was my assistant throughout this sampling. We then moved back east to Illinois, where we sampled at Tower Grove Cemetery, which is a disturbed site. So it's repeatedly mowed. And Triodanis biflora was the only species present there. And while abundance is comparable to other sites, species richness is very low. So we only had one genus present there, and it was the syrphid fly species. So 2 different syrphid fly species, Toxomeris. So like I said, when I was introducing my methods, we did this other haphazard sampling, and we generally did this across the states where we sampled. What we found is that the pollinators that were present in this haphazard sampling were very similar to the ones that we found when we were doing our standard sampling. This means that, well, we can sort of infer that these are the 2 functional groups that make up the main pollinators of Triodanis. And that's sort of like the key takeaway there. So to wrap everything up, we found that generalist pollinators were present across all 4 species of the Triodanis that we sampled. And it was repeatedly represented by these 2 functional groups, so flower-feeding flies and small native bee species. And then with differentiation between Triodanis species, obviously our sampling was pretty low coverage, even though we were doing the standard sampling, but we still found that pollinators didn't differ due to breeding system variability between these different species. Oftentimes, and as I said, oftentimes they were sharing pollinators when they co-occurred. And then does pollinator assembly differ spatially across the ranges that we sampled at, so across the Midwest and Texas? And what we found is that no, trends generally agreed across spatial scales. And then while abundance and diversity was varied dramatically across the landscape, our findings seem to confirm maybe some of our fears when we're thinking about pollinators, and that these human degraded or disturbed sites generally hosted less diversity and were possibly just not great habitat for native bee species. And so with that, I'm going to go ahead and jump into the next chapter of my thesis, which was a citizen science project that I employed here at SIU. So in this second chapter, we're going to talk about a new generation of citizen scientists, so these being college students, and this voluntary project that I applied in a college classroom setting. In the introduction and background, we're going to talk about what citizen science is, if you're not familiar, and why the advantages to applying it in a college classroom. We're then going to jump into my project design and how I implemented this project. And while I do that, I'll talk you through some of my analysis and results. And then we're going to wrap up the key takeaways from this project. So what is citizen science? Citizen science is generally the use of non-experts to gather scientific data. And you may know it as community science. And so why should we apply it in the college classroom? A college classroom is sort of its own community. College students are the newest generation of citizen scientists. They're newly autonomous. They're starting to pursue their own interests, and they're starting to own their knowledge. So instead of catching citizens when they're out in the community, we could catch them early here in the college setting. So I think there are some real advantages to using citizen science in the classroom. One of which is that it's generally self-directed. Projects are decentralized, so students can work on them at their own pace. And we know the importance of self-directed or decentralized learning from the pandemic. So we shouldn't necessarily stop what we were doing then, and we can adopt some of these projects while we're now physically in the classroom. It also has the possibility of democratizing science. So there are some college educators in the room. I think we generally think of science as a process, and there's lots of trial and error. But for students, I think this is a black box. And it may be hard for them to understand this trial and error process. You don't become an expert overnight. So citizen science has the ability to replicate how you would teach yourself a skill outside of the classroom with this decentralized learning. It can also engage diverse groups. So generally, in the past, citizen science has engaged a pretty narrow demographic, and it generally skews older. So in the college classroom, we have students who are generally much younger, and we can engage them. And then we can send them out into the community with these skills for creating projects to make change. So overall, there's been a push to standardize research in citizen science. The Cornell Lab of Ornithology has been really good at doing this standardization, but they've done it for community projects. And within the last couple of years, there's been a push to standardize this research in the college classroom as more people are applying it there. So Vance Chalcraft put together a summary of some of the reasons why college educators would want to employ citizen science. And for this Vance Chalcraft paper, there were very few of these evaluations that college educators were looking at if we compare them to their motivations. So as educators, we can think about some of the reasons why we pursued this course. So having students be excited about science and promoting lifelong learning. So the motivations for the Vance Chalcraft paper were very diverse, but they generally fell, both evaluations and motivations generally fell into two broad categories: social awareness and self-perception, and then scientific inquiry. I broke these broad groups down into three and identified them as my learning goals or learning outcomes for this project. And so these were self-identity and motivations, self-efficacy, and skill growth. So my goal was to implement this project briefly in the fall of 2022. And so I implemented this project in 4 different classes in the plant biology department. They ranged from lower level courses or introductory courses to 400 level advanced courses. I did this with the cooperation of the professors of the classes, obviously, and then with the approval of the internal review board here at SIU. Generally, all of these classes have scientific observation as part of their core curricula. And so it seemed pretty natural to employ a citizen science project there that was promoting these skills. So to introduce this project to the classroom, I went in person to talk about citizen science generally and tell students where they could find the workflow for this project, which was on their learning platform D2L. This project overall was voluntary, so we couldn't ask students to participate in this project, but we could motivate them by talking about the importance that this work could have. So they were told where their workflow was, and then they were moved to a citizen science platform, iNaturalist, where they could interact with some of the other parts of this project. One of which being a pre-survey. So we wanted to make sure that we were evaluating outcomes or that we had a baseline for outcomes before we we directed students into the training module. And so the training module was based around pollination ecology, and it covered some broad topics that I'm going to talk about directly. Students were then asked to interact with a post-survey. So the pre and post-survey had the same questions. So students were interacting with the same survey both times. The training module started with these inherent mutualisms between plants and pollinators. And this seems really basic. It's something that we've heard about for a really long time, but there's research that shows that primary education often misrepresents what are really complex ecological interactions. So we wanted to make sure that every student that interacted with the module had this baseline knowledge. We then moved on to how students could identify floral visitors versus pollinators. And we talked about how they could identify those floral visitors and pollinators. We then moved into more complex topics, such as pollinator assembly and pollinator syndromes and plant communities generally. And then we directed students through how to conduct observation-based research. And this was for observations of pollinators and plant communities. We then taught them how, if they wanted to take these observations, how they could contribute to the scientific community by posting these observations on citizen science platforms like iNaturalist. And then it wrapped up with the importance of these observations. So what one individual could contribute to our scientific understanding of plant-pollinator interactions just by posting a picture on a platform like iNaturalist. So like I said, there were 2 different surveys, and we took just some standard demographics at the start of the survey. And then we also asked students to self-identify themselves as STEM or non-STEM. They then had a free answer question where they just put their major down. And what we found is that most of these students were misidentifying themselves. If we compared what they put down as their major to their answer for identity of STEM or non-STEM, students were repeatedly misidentifying themselves. So only 3 of the 27 students who put down or who self-identified themselves as non-STEM were actually students that at the end of their coursework here at SIU would graduate with a Bachelor of Arts. And of these students that misidentified themselves as non-STEM, most of them were majors housed in the College of Agricultural Sciences, so CALPS. And a majority of them were forestry majors. And so forestry major here at SIU teaches you how to manage habitat and species. And so it's sort of this double layer that we have with talking about these cornerstone ecological interactions in the module, and then having these students who could go on to be land managers misidentifying themselves in the sciences. We also asked students about their current participation in citizen science to establish a baseline, and most students just said that they were interacting with citizen science as a student. So because we wanted to understand this baseline, we got the baseline essentially. I'm unsure how to interpret that result. But we also asked students about their motivations for interacting with citizen science. And while they answered that they were interacting with it just as part of the student curriculum, they also answered equally that they interact with citizen science because they like to enjoy and preserve nature. And it's important that we understand these motivations if we're thinking about adopting these projects in the classroom, so that we can really hone in on what gets students excited about this learning. In other parts of our survey, we used polar and Likert scale questions to look at the 3 broad learning goals that I defined. Polar questions are just yes or no questions. And we were looking for a switch from no in the first survey to yes in the second survey. So we were looking for a positive response from the first to the second. And what we found is that overall, students were switching from no to yes. So for overall participants, we also, so we use this, these questions as sort of a proxy for self-efficacy. So one example question would be, have you participated in observational study? And so we had a switch from no to yes for overall participants. The rest of the survey tested all 3 of the broad learning goals. And this was a sort of a huge chunk of questions. So students answered 23 different Likert scale questions. And if you don't know what a Likert scale is, you really do, because you probably interacted with them. If you've ever interacted with a question that has told you to rank something like, oh, I'm likely to do this, or, oh, I'm really unlikely to do this. And so it was broken down into 3 different sections, one being agreement. So I currently have the skills to blank. And so we were looking for students to rank themselves as, yes, I strongly agree, I definitely have these skills, or no, I don't have these skills. We used this agreement as a proxy for skill growth in scientific inquiry. We use likelihood, so how likely are you to repeat something, as self-efficacy. So if you feel like you're effective at something, you're probably more likely to do it again. And then importance. So how important are these ecological interactions that we taught you in the training module, how important are they to you? And we used this as a proxy for self-identity and skill growth. And what we found is that for overall students, we had a positive response. So it's a 4-point Likert scale, 1 being a negative response, 4 being a positive response. And for overall students across these 3 broad question groups or learning goals, we found a shift to a more positive response. We then broke the respondents down into self-identified STEM and self-identified non-STEM because we thought that was really important. And so when we looked at the self-identified non-STEM, we also found this positive shift across all of these different learning goals. For our self-identified STEM majors, we found very little improvement, and that was because they were already ranking things really positively. So they felt they understood maybe the importance of these ecological relationships. But where they didn't rank themselves positively are these scientific skills that we looked at in the first section for these Likert scale questions. So one question that students across the board, so non-STEM, self-identified non-STEM and self-identified STEM was define a pollinator. So, they did show overall shift to a more positive response across groups. So overall for define a pollinator, apologize. But for other questions across groups, self-identified STEM didn't really show much improvement for teaching others about pollinators. So maybe they feel a little less confident in a skill like this. And then also for overall students, they didn't really show much improvement for ranking of ecological services, which means that maybe our module just didn't really cover it enough. And so they don't really understand the importance of these relationships. So to wrap up, these projects that integrate citizen science and higher learning can have a positive impact, even if they're brief and voluntary. We had ranking improvement for learning goals across overall students, but without testing the effectiveness of these projects, we miss opportunities as educators to advocate for their presence in the college classroom. And if we miss this opportunity, then we also possibly miss funding. And we're really doing ourselves a disservice because I think that they are truly effective when employed in higher learning. And I will be presenting this project in May at the CSA conference in Phoenix, which I think is really exciting. So we're going to wrap it all up into like a nice, neat bow. In my first chapter, I think I stressed the importance that natural history projects can still improve our knowledge of these understudied systems. I think we discredit these projects because they are descriptive, but we understand that there are understudied organisms in both plants and pollinators, and we don't necessarily understand their interactions. With the increased anthropogenic pressures on these interactions, it's even more important that we establish a baseline as we continue to see this rapid change across landscapes. So I think that sets up the importance for training and familiarization of students in general observations. This doesn't necessarily have to be for pollinators, but giving students the skills to observe organisms in a scientific and standardized way allows them to go out into the world being better scientists even if science isn't what they pursue outside of college. And then there's also this continued importance to look at self-identity for college students in the sciences, because maybe they just don't see themselves there. Like they're there, but they don't really see themselves as scientists. And then we can have this greater impact by sending these students out into the world more prepared. All right, before you clap, I want to give a big thank you to my lab. They mean a lot to me. And I was so lucky to have Dr. Weber as my advisor. I had to trust the process. I didn't always trust the process, but you do have to trust it. It works. And in that process, I became a better scientist. There were so many labbies that had a hand in my research, and I want to give a big shout out to Taylor Simmons, who's in the crowd, and Sarah Lukowski, who skipped my presentation to go to class. Lame. Because they braved so many different things, like how hot it was last summer. It was so hot. And maybe like things like tornado watches, and my driving and my choice in podcasts. They had to endure all of that, but they did so with a smile. And for that, I have to thank them a lot. There were also other lab students that had a hand in some of the bench work that we did. So Julia Beckner, Josh Bellows, and Taylor McKenna, who are all here today, you all get a shout out, because without your help, I would not have been able to complete this project. And then there were many other lab members that I won't go on to name that have helped just prepare, just be there, just be there for questions and other things that come up in research. I'd also like to thank my committee members, Kurt Newbig and Sidonia Sipes, Dr. Kurt Newbig and Dr. Sidonia Sipes, for their expertise and their encouragement in my research. I want to thank my good friends here at SIU for your support, and then also all my far-flung friends who are here on Zoom supporting me. You all get a big shout out, because without you, I probably would have gone crazy a long time ago, but your support has really meant a lot to me. I want to thank Eddie Chavez for always doing the hard things with me. And then there are many different field managers that I'd like to acknowledge that helped me with paperwork or helped us find pollinator populations. They made my work a lot more efficient and effective. And then I also need to thank my funding sources. So the Superb Scholarship, I would not have been able to do this research without it. The generous grant from the Missouri Native Plant Society, and also the grant I received from the from SIU GPSC. And with that, I'm gonna wrap things up.